Thank you so much for attending the 2021 UA World Innovation Summit Spring Conference. I am the host today, Peng Zhuyu from MIT. Today, it's a great pleasure to have invited Professor Jean-Marie Lin as our valued guest speaker. Professor Jean-Marie Lin is a Nobel Prize laureate in chemistry and pioneer in the chemical basis of molecular recognition. As an author of more than 900 scientific publications, Professor Lin is a member of many associations, academy, and institutions. He's also a recipient of numerous international awards and honors, including selection as a foreign associate of the U.S. National Academy of Sciences, the Paracelsus Prize for Swiss Chemical Society, and medals from Charles University and Polish Academy of Sciences, and so on. Over the years, Dr. Lin's mainland research has expanded from the studies of molecular recognition to the definition and exploration of new fields in chemistry. Since the winning of Nobel Prize, Professor Lin remains dedicated to his research, but is also committed to reaching out more broadly. He's interested in more scientific approaches to approach problems and also trying to convince the public about the value of science. During the speech, if you have any questions, please feel free to type them in the Q&A box below. At the end of the speech, we're having a Q&A session. Now, without further ado, please join me to welcome Professor Lin for his talk. Professor, you can show the screen. Yeah.
Okay. You see it?
Yeah, perfectly.
Okay, very good. So thank you for the invitation to this rather special type of organization. I have given many talks because of the confinement, of course, we have the virus and all that. So everybody is hanging on video exchange, and we are all becoming zombies. But this is an interesting one because you have several universities from different regions. I see they are mostly Anglo-Saxon. I think you should also look to Europe. There's a lot of things going on in Europe also. I recommend that. It's not just the US and the UK. Okay, that's what I usually say. We can talk about that a bit later in the discussion. So it's an innovation summit, but I will not talk about much about innovation. I will just give you a sort of a story of science, very broad, but centered on chemistry. The core will be chemistry. So the title is Steps Towards Life. I have added and thought. You will see why. And I claim that this is based on chemistry because we are all made of molecules, as we are. So we have to start very early in the history of our universe. The universe was born about 13.7 billion years ago in a Big Bang. And at that time, the universe was extremely hot. There was no chemistry. There was energy, it cooled down, particles formed, particles associated to give atoms, and then atoms associated to make molecules. This is the age of physics. No chemistry at that time. It is much too hot for chemistry. But when it was cold enough, then the chemistry started around here in our universe. That's the age of chemistry which starts. Molecules became more and more complex. They started to associate, to make aggregates. These aggregates made sort of vesicles, made sort of containers. And something that happened at about much closer to our time now, which led to the formation of living organisms. So non-living matter changed into living matter by a mechanism we don't understand yet, but we will certainly understand. And life on our planet, Earth, started about here. This stage of biology. But it didn't stop there. Evolution of the universe continued to become more complex and generated a property which is, in my opinion, even more important than biology, than life. This property is thinking. This is why we are here. This is why you do what you are doing. That is why you go to universities. That's why you have a job, which is in innovation or in research. This is thinking. And this is the steps towards life. From the Big Bang to physics, then developing into chemistry, developing into biology, and finally generating this fantastic property, which is thought. And we'll come back to that. I show here at the end of the screen this sculpture, which you may see. That is the thinker of the French sculptor Auguste Rodin. This is the person sitting there and thinking. This is our attribute. Notice also that we are a point in space and in time. We are not very specific. We are not something special, one would say. Although in a moment you will see that it is something very special there. But other organisms in other regions of the universe may well have the same property. And I'm quite sure that there is life in many places and thinking in many places. So we have to be modest, but at the same time proud, and you will see why. So this is a slide which this looks empty. It's not empty. This is what the universe was, dark. 68% dark energy, 27% dark matter is what physicists, cosmologists now think the universe is made of. 95% darkness. Not very clear, all this, but there is 5% visible matter. And this is the matter that matters. We are part of this 5%. Because we are there, because we are 5 of this, we are here in this visible matter, we can think about the universe, and we will see in a moment what happens. Now, this evolution of matter, or visible matter, one should say, is something which seems to be linked as a function of time with complexity and information. In other words, as the universe became more and more advanced, more and more complex than a long time, information made things organized. And you will see in a moment that this evolution towards highly complex matter, very informed matter, is also the evolution from the divided matter, the particles, to the condensed matter, bringing these particles together to make atoms and molecules. Then they get organized. Then life appears at some stage, thinking appears, and maybe beyond that, there is something else. I cannot tell you what it is, because it's very difficult to think beyond your own thinking. But there could well be some other higher forms of activity, of thinking, of property. And that is the evolution of matter. And I might say it looks like there's a pressure of information to push it this way. So the big question we then can ask, what is it? It's quite simple. It's the biggest question, but it's simple. How does matter become complex? How come that from the Big Bang, human beings evolved? Now, this is a very complicated question. How is it possible to go from an elementary particle to a thinking organism? And maybe there are even higher forms of complex matter. To answer this question, mankind on our planet, this very small piece of dust in the universe, at a point in space and in time, mankind invented science. Let me just look at 3 fields. Physics deals with the laws of the universe, the laws which run our universe. These are the fundamental laws, and everything else depends on these laws. Biology deals with the rules of life. These are not laws. The laws are already there, but they are the rules, how it functions. How does a living organism function? So what is chemistry doing? When people ask me, what is the field of chemistry? I tell them chemistry is a fantastically important mission to establish the bridge between the general laws of the universe and the appearance, the coming out of a living and a thinking organism. So chemistry builds the bridge towards complex matter from the general laws to specific expressions of these laws in more complex states of matter. Now one can claim that the answer to this question is residing in one word, which is self-organization. I mean by that our universe has such a structure that it will necessarily get organized. It is not an accident that life exists. It is a cosmic imperative. Self-organization is part of the structure of our universe. And this then leads, of course, to this evolution towards more and more complex forms of matter on our planet, this small place in our big universe, and this thinking organisms. Now, self-organization has to be considered in 2 ways. on the large scale, cosmic self-organization results from the structuration of the universe through gravitational forces, Einstein's forces, which act on the inhomogeneities and the rate of expansion after the Big Bang. This is not our field. This is not what we are worried about today. Complex matter comes from the organization of matter, these things which form by condensation, by formation of particles, atoms, molecules, and so on. And this gives rise to a sort of a random structural combination, which then happens some to be better than others, better meaning thermodynamically preferred over one rather than another one. And this is the matter we are interested in because we are part of it, molecular matter. So the chemical evolution of matter started much before there was only Darwinian evolution. Darwin only came about when it was life, when life was around. Before that, there was much more evolution coming out, which was the evolution before life, prebiotic evolution, which is an evolution which is evolution of non-living matter. And it's obvious that there must have been a non-living evolution because it's very difficult to imagine to jump from a particle to life. So matter was becoming progressively more and more and more complex until it reached the threshold of developing what we call a living organism. Now, this biological Darwinian evolution is then a follow-up of chemical evolution, and I could even say that, in fact, Darwinian evolution is still also chemical evolution, as you will see in a moment. And all the people who do biology here, they know that very well, because biology is based on molecules. So it's an evolution, which is a chemical evolution. Now, chemistry is defined usually as that part of science which deals with the structure of matter, how it is built, and the transformation, how you can transform objects of matter into one another. That is why I'm a chemist, because chemistry can take pieces, take objects of matter, and make other ones out of it. And this is a fantastically enticing, very exciting and powerful type, sort of even a power trip, if you want, of what science can do to transform the elements, the structures, the components of matter into one another. Now, over the years, mankind had studied what is around us, what is this matter we are made of, and what is made of this. And this, of course, was from the beginning of civilization, from the beginning of mankind. This was a big question. And progressively along the times, one discovered that there were sort of bricks of matter, which then were analyzed and were put on a sort of put together, but that had no order. It was difficult to see why this thing with this sort of bricks one found, what are they doing? Is it just chaotic? Is it just anything? Or is there an order? So in the middle of the 19th century, a number of people proposed ordering these bricks of matter. The one which will give the best representation and which the one who really led to what is now what we know about is Dmitry Mendeleev, a Russian chemist, who published in 1869 this paper. You should look at this paper. That is one of the most important papers ever written in science. This is a paper which has changed the world, has changed science. This paper tells us that one can organize the bricks of matter, the elements, according to their atomic mass. And this is an organized table. This is you see it here. So the title of the paper, which was published in 1869, was in German at that time, on the relationship between the properties and the atomic weights of the elements. And he classifies these elements. This classification is down here, as you can see. For the chemists, they find here what you know in the presently. There's beryllium, boron, carbon, nitrogen, oxygen, fluorine, and so on. There is beryllium, magnesium, zinc. There's calcium also somewhere, and so on. So all this is now a table, and this table is the table which has given rise to the present day one, which I can show you, which is called the periodic table of the elements, which is here. So you have to look at this. This is one of the most important pictures you can ever see. This is a table which shows all the elements of visible matter in our universe. I repeat, because it has to go into your brain and to be engraved. It is the table which shows you the bricks of matter which make visible matter in our universe and which exist everywhere in our universe, and there are no others, because this table is full. Because this table has nothing, no space between the elements. Between lithium and beryllium, there is nothing. Between boron and carbon and nitrogen, oxygen, there is no space between. They are just following up, counting the number of electrons and number of protons. So this table is complete. And for instance, a carbon atom will be the same on our planet or billions of light years away. This is something you have to realize what that means. That we, this little animal with 2 legs, 2 arms, but with a head and a brain, has been able to put into a table the bricks of our universe. And that's the playground of chemistry. Chemists play with these elements, recombine them, connect them together, and make this infinite variety, diversity of what can be done on the basis of physical laws, because you cannot just do anything. Physical laws control and determine what you can combine and how you can combine it. But the diversity creation, the creation of new entities, that's the hand of the chemist. So from the atom to the molecule, matter became more complex. That is molecular chemistry built up. It started when? We will see that in a moment. So molecular chemistry is linking together atoms through strong bonds, which for chemists and for all of the scientists who have a little bit had chemistry are called covalent bonds, strong bonds between the atoms. Now, milestones in molecular chemistry can be defined and could maybe arguably look at some other things, but I would say it is sort of accepted that the first step was the production in the laboratory of urea, which is present in the living organism, by Friedrich Wöhler in 1828 in Germany. He showed that you can make urea from ammonium cyanate. Ammonium cyanate is a non-living matter entity. Urea is in living matter. So he did 2 things which are very important. First of all, he transformed ammonium cyanate into urea. That's a chemical transformation. Second, he destroyed that myth, which was called at that time the vital force. People were thinking at that time that you could not make anything present in the living organism without the help of a magic force called the vital force. By converting ammonium cyanate into urea, he showed that vital force does not exist, it's not necessary, and it's a continuum. This is also philosophically very, very important step in that respect. Now let's jump this 1970, middle of the 1970s, this very big molecule, vitamin B12, was synthesized by the combined efforts of 2 groups. One the director of that group was Robert Burns Woodward at Harvard University. I was a postdoc with him. I worked on the synthesis at that time, 1965 and so on. And the other group was led by Albert Eschenmoser at the Polytechnical Institute in Zurich. These 2 combined their efforts together with 100 co-workers or something like that over the years to make these molecules from scratch. Scratch meaning from the atoms, putting the atoms in the right place, in the right position in this molecule to obtain vitamin B12 along about 80 to 90 steps of man and woman years. Now, of course, this shows how powerful molecular chemistry is. Making molecules, making materials, finding new processes, finding new reactions. And this has continued in the last 50 years or so, of course, and it will continue. So molecular chemistry is alive and is very well progressing. However, we have to ask a question, is there something else we should be interested, we should look at? Let me introduce it by biology. Here we have a cancer cell and 2 killer cells. The killer cells have as mission to recognize, find out the cancer cells, not make an error, of course, and destroy them. but they should know that the other one is a cancer cell and not a healthy cell, not to get problems. Here also, the blue dots is the HIV virus. The red thing here, which is artificially colored, is a white blood cell. When the HIV virus hits the white blood cell, it can penetrate. How does a virus know that it has reached its goal? So something happens between these big bodies, which are all made of molecules, of course, for the chemists, biologists, and so on. These are membranes which define the objects in question. These membranes are made with molecules, and they contain given molecules which sort of characterize what they are. So the cancer cell expresses some molecules which says, I am a cancer cell. And the killer cells will recognize that and then have the job to destroy it. So let's look at this. This is a point that we have now to deal not with isolated molecules, but with what happens when molecules get together. And this means that beyond molecular chemistry, there is what we have called, I introduced this name in 1978, supramolecular chemistry, which is a chemistry line beyond the molecule, which uses interactions which are not this covalent type of interactions, and which have three main properties which have been studied over these last 50 or 60 years: molecular recognition, molecular reactivity, how do molecules react with one another, and transport. Recognition is the basic process. How do molecules recognize one another? What do they do to do that? But I will give, of course, a very simplified, enormously simplified image, but it's an image which gives you a feeling. Molecular recognition requires interactions for binding. You need some physical interactions to bind together. If there is no binding, the objects ignore each other. But there is another property which is very, very, very important. That's information. You cannot have recognition without information. So there must be information in the molecules. And there is a way for molecules to bind selectively to one another. This can be simply defined as a complementarity in geometry and in interactions, a type of interactions. Like, as you know, plus attracts minus and plus repels plus, things like that in the extremely simplified way. But I can already then show you that, in fact, in the 19th century, again, a chemist has already proposed an image for that. This is Emil Fischer. And all the biologists and the chemists must absolutely know Emil Fischer. If they don't know him, they should go back to their books. So Emil Fischer published in 1894 a very famous paper where he studied the action of enzymes on their substrates. And he said that in order to act for enzymes on the substrate, they have to fit together like Schloss und Schlüssel, like a lock and a key. It's a very mechanical way of looking at things, but basically it is correct. The lock and the key, they fit together and they sort of recognize each other. In addition, when you have the right key, you can open the lock, you can open the door. So you have a function which comes later on. Notice that we are very proud of the fact that he got his PhD in our university in Strasbourg in 1874. After that, he went to Munich. So that is Emil Fischer. At that time also, Adolf von Baeyer. He was the research director of Emil Fischer in Strasbourg. Okay, so the most important information system, arguably much more important than any computer, much more important than anything, is this one. What makes human beings, what makes living organisms on our planet? This is a code written by 4 letters. There is a molecular storage of the information in the genome of all living organisms. And these are just 4 letters. Adenine, chemists have given names to those kind of objects. That is just, they could have taken other names. They could have said 1, 2, 3, 4, but they call it adenine, guanine, thymine, cytosine. And they are rather simple, trivial chemical groups, as you can see here. These are trivial things. But these are letters sticking on a long, long strand, which is rather simple also, of phosphates and sugars. They're wrapped, and 2 such strands wrap around each other, as you can see on the right-hand side, into the famous double helix, which everybody now has heard of, I guess, in the world. And so this is the storage of information. But it's not enough. You have to process it. What's the mechanism for processing? The mechanism for processing is supramolecular, is interaction between these molecular systems. And it happens by either 2 points of interaction, like the pairing up of adenine and thymine, or uracil, is just a slight derivative, with 2 points of interaction represented by these little dots, or by 3 points of interaction between guanine and cytosine with these 3. So it's just a binary simple, a very trivial binary system. 2, 3, 2, 3, 2, 3. That's the way you read and process. So you can, if you think about it, is really a bit, it's a bit disappointing, I would say. You write the genome of all living organisms, the difference between a tomato and an elephant, with a series of 4 letters, which just undergo pairing. Why is it this? Possibly that is if sufficient, you don't need more, and probably evolution has tried other letters, more letters, but it was perhaps very chaotic, many errors formed. And so the simplest remained the best. So I claim then that chemistry is an information science. It's the science of structure of matter, of transformation of matter, but also the science of informed matter. Information is stored in the molecules and is processed by touching, by contacts between molecules. So it's the science of supramolecular chemistry is the science of populations, of what happens. It's a sort of a molecular sociology. What happens when molecules get together? So molecular recognition then, because it's so fundamental, of course, has been studied very extensively by many, many laboratories around the world, in all countries. I have a lab in Canton, in Guangzhou, and of course I have many Chinese colleagues. I know them very well, and so many excellent laboratories work on that everywhere in the world. So it's a question. Let's see if we simplify. It's a question of playing with locks and keys. That's just. This is, of course, that's very, very simplified, but it's a question of locks and keys. And many laboratories have contributed to that. Now, this being said, let me quickly have some applications. Now I jump. I don't talk about the rest because too much. I could talk for a month. But let me just mention a few applications which come from our work, but there are many others, of course. So as you know, basic research is acquiring knowledge. That's what we try at the universities when you do our PhD, when you and so on, when you do our research. And applied research is applying this knowledge. This is as important because without applying the knowledge, you have no applications. Now, let me just show the first application of supramolecular chemistry is, of course, drug discovery. A drug is a molecular key for a biological lock. And understanding molecular recognition is fundamental because you want to make keys which are very, very, very selective to act on one given lock in the organism and not on others. So this is why the most important property is molecular recognition. Without molecular recognition, we do not exist. In our body, as we are sitting here, as we are listening, all the molecules do their job all the time in the right way, at the right speed, in the right place. Now, we developed also an apparatus we developed a label for medical diagnostics, this kind of unit here, which shows that UV light is absorbed by this entity. And in the cavity of this molecule, which you see here in the middle, there is a europium ion which emits a red light. And this is a sort of a label which then can be followed. And this has led to an apparatus for immunoanalysis, which was developed by a small French company, now a bigger one, Cisbio, by Dr. Gérard Matisse. Another property which is linked to recognition, but which is also transport. This is gene transfer. What is this? I think I talk about it because it will become more and more important in the future. It's already important. It is the process by which a piece of DNA, a gene, is is led into a membrane into a cell through the membrane by synthesizing synthetic vectors. There's also a viral approach, which I don't want to talk about, but it's an approach by which a piece of DNA, a gene for a given protein, is introduced into a cell. Then it goes to the nucleus, it makes its RNA, and then out of that comes the corresponding protein. Now, here, just a general remark, but all of you who are listening, I suppose, are scientists. So for you, it must be obvious that gene transfer, of course, is important for gene therapy, which has been done, but also for biotechnology, for making genetically modified organisms. When I give a general public lecture with people who are not scientists, I insist on the fact that genetically modified organisms are here to stay. We need them. People are getting afraid of them. That is stupid. That's crazy. There's no reason to be afraid. We can control it, and we need genetically modified organisms. And we will do it, and we have already done it. Finally, supramolecular materials are materials which are developed on the basis of supramolecular chemistry. And let me just show you one thing here, which shows the jump, the enormous jump which can come from basic research to applications very far off from what you did initially. This deals with a story we started in 1990. We started by making supramolecular polymers, and we published the first paper in 1990 on supramolecular polymers. Then a small company, Xeltis, made them biocompatible and developed out of them polymers of supramolecular nature, which were used for the cardiac reconstruction of children having a serious, very serious, severe cardiac malformation. Just look at the jump from initial polymeric studies, supramolecular chemistry, to something which you introduce into human body of a child. Here is the first child which was reconstructed, whose heart was reconstructed. This is young Dominica in 2013. She was 4 years old. That was the first implantation by Professor Leo Bokeria, the pediatric surgeon at the Bakulev Scientific Center for Cardiovascular Surgery in Moscow. And this is here, she is already here. This is 3 months after implantation. But now she's, of course, since 2013, she is now almost 10 years older, and she's fine. Many other children have been reconstructed, so to say, and successfully treated. And the surgeon has a nice smile. I think I agree with him. It's fantastic to have been able to do that. So the company also developed, just to tell you, also pulmonary heart valves, which are shown here. It's more complicated because they have to move and all that, but I don't want to get into all this. This is now this is done and it will continue. But for us, it was a fantastic satisfaction, the fact that from purely research work where we had no dream, no idea that we could one day, this could end up in the heart of a child. The other property of interest is that molecular self-healing, molecular supramolecular polymers also have the property, when you cleave them, to reconstruct. Here you have a film, you almost do not see him, very nice transparent film, which was done by one of my Indian postdocs. You see his fingers here. You cut it in two, then you superimpose the two ends, you press with your finger, and you can stretch. It sticks. This is in 5 minutes, less than 5 minutes, in fact. You cut, you superimpose, you press, and it sticks. And this type of properties, self-healing materials, are of course also great importance for development of material science. Now, let's go back to the main thread of what I said. I said that self-organization is really the driving force in our universe. So once you understand better supramolecular chemistry and molecular recognition, you can go a step further than just making locks for keys and keys for locks, but letting these things assemble by themselves. That means introducing the notion of self-organization in chemistry, But this is spontaneous formation of objects, but directed under the control of molecular recognition. Spontaneous, but information controlled via molecular recognition patterns. And an example is a virus. Now, as you know, a virus, you look, it's biology. Okay, yeah, it's biology, but a virus is just chemistry. It doesn't live. A virus is not really living, as all the biologists who listen to my lecture know. It's just at the brink of death of non-living and living matter. But let's just have a quick look. I chose the first one, which was understood, tobacco mosaic virus. It is made out of 2,130 protein subunits, one molecule of the RNA, the genetic code of the virus. And as you can see, this 2,130 protein subunits, they assemble, they go around the circle like this piece of cake you see on the left-hand side, and then they assemble and they form this tower with a hole in the middle, and the RNA is sticking in this hole. This looks like magic, but it's nothing magic at all. We understand every step. It's just a molecule which has the right shape, the right recognition patterns, so that they assemble on the basis of these recognition interactions so as to generate the final object. It is just built in. It's organic chemistry, structural chemistry, physical chemistry, supramolecular chemistry, and so on and so on. So it's a programmed chemical system. The program is written in the molecules, and then it runs, and the output is the virus particle. So these programmed chemical systems, there are different ways of looking at them, and the molecular program, and this operation is supramolecular, and it goes through molecular recognition algorithms. Let me just give you some examples of this. Just what can be done in the lab, because of course people and chemists said, okay, all right, that is interesting. That happens in biology, but can we also in the laboratory make pieces, objects, components, which will spontaneously assemble to generate a complex object using molecules which can bind metal ions. The molecules are the bricks, the metal ions are the cementing units, the connections. One can make an enormous variety of, let's call them objects of supramolecular nature. I give you just a few examples of what has been done, but of course, hundreds and hundreds and hundreds have been made. The first ones we made are these ones, an artificial double helix and an artificial triple helix. They have nothing to do with the natural one. It was just to demonstrate that we can make double helices, we can make triple helices of totally different nature from DNA in the laboratory. It was just a game to show we can do it. This is a grid where molecules in blue and molecules in red are held more or less perpendicular by binding 9 metal ions. These are silver cations. And of course, 3 times 3 is 9. So you have 3 intersections along each strand, and then you get an object which is a grid which has 9 well-positioned metal ions. You can make a sort of a cylinder where you use flat molecules in blue, linear molecules, straight molecules in red, and metal ions to connect the whole thing. In this case, 4 flat ones, 3 linear ones, 12 connectors. These 19 components, they go together spontaneously. You mix and you get. And these are some others, just to give you some nice pictures, that's all. But there are many people now, and there are much more complex objects which have been made also by the same type of design. So this can also be of interest. I want just to point that out for future nanoscience and nanotechnology. Nowadays, when one makes our nice objects, the devices which you all use, we make them by construction, so to say, by fabrication. But one can also think that maybe one will be able to design systems where the system itself selects the pieces it needs to build itself up on the basis of molecular information and supramolecular interactions. This would be a powerful complement, maybe not an alternative, but certainly a complement to the present-day nanofabrication and nanomanipulation, and is going from fabrication to self-fabrication, which is, of course, the ultimate form of fabrication, letting things build up themselves. Now, molecular chemistry and supramolecular chemistry can be performed by design. You take pieces which contain information, you put them together, you can program your system, as I have given that example virus, but this is the other ones with these metal ions holding together molecules, it's also the case. And this then leads to the generation of what you may call functional entities of either molecular or supramolecular level. Okay. But there's another approach, a step beyond. Another step in complexity is now to ask the question, what happens if the system is able to do it by itself? In other words, beyond making things by design, there is letting things make themselves by selecting the right components from a diversity of components. Now, this is what we are doing now. And many labs have joined the field, but I cannot get into details. I need another hour for that, but I cannot go into details. Let me just say that it means that you have to work with highly diverse mixture of objects, which then on the basis of recognition and interaction select themselves. The system has to be dynamic so that the first encounter between two objects does not lead to something which sticks so well that it cannot dissociate anymore. Maybe the first encounter is not the best one thermodynamically. So one needs diversity of building blocks, dynamics in the association of these building blocks, and leading, therefore, to a selection of the right building blocks. Now, this led to a chemistry which we call constitutional dynamic chemistry. I'm sorry for the complicated name, but it means something rather simple. It means that the constitution, the way the object is built, is dynamic. In other words, it is formed of pieces which can fall apart, dissociate, and then reassociate. But along this process, if the conditions change, it can reassociate differently, or it can maybe kick out some of the pieces and take in some others. So it leads to an adaptation of what happens, of the surroundings, and we call that adaptive chemistry. It's a chemistry where the chemical objects can fall apart and reassemble, but differently, depending on what happens, depending on whether something has happened, like the medium has changed, like something has been added to it, and then the chemical system adapts to it. So this is what we are now doing. Mostly I had some example, but I took it out because it would have been too long. And maybe I give another lecture sometime. Okay, so this is the adaptive chemistry, a chemistry which is now very much in the development. So, wait a minute, I left one of them. So I show it quickly, but let me not go into details. Let's just show what it can do. These are now two dynamic polymers, okay? And we superimpose them by an end, by a corner. A and B are connected, A prime and B prime are connected. But the connection is dynamic, they can break apart. So when you superimpose, two new combinations can be generated there. A can combine with B prime, and A prime can combine with B, okay? These are two new combinations which do not exist in the initial entities, the initial polymers. They can, one of these can be colored or fluorescent. So let me just show you what happens. That's a cat, the head of a cat, and the head of the cat and the ears is AB. The inside of the ears, the eyes, and the mustache is A prime, B prime. You heat, and now you get two new combinations. And it happens that one of these combinations has a color and the fluorescence. You get color, you get fluorescence only where it is superimposed. That proves that indeed the recombination works, that these new systems, these new combinations have formed, and it shows one of the powers. It's just an illustration of this adaptive chemistry, this dynamic chemistry. So we have come a long way in a very short time from molecular chemistry to supramolecular chemistry. Then it gets organized. It has to be dynamic, otherwise it's dead. Death is now no dynamics anymore. Life is dynamic, adaptive, and these are ways towards living matter, thinking matter, and so on. So let me finish with some general considerations, because there are lots of students here, and I think it's good to talk about these things. First of all, I want to make a case for chemistry. Chemistry has not just to discover what already exists, but to create what does not yet exist. And the book of chemistry has to be written, not only to be read. Of course, we have to read it. We have to see what we are made of, what trees are made of, what stones are made of, and so on. But we can make different ones which have not yet been made, combinations that have been made. So the score of chemistry has to be composed, not only to be played. So chemistry has a lot of creative power, and I want to compare it to art by taking again an example from Auguste Rodin. This is the hand of the artist expressing out of the stone a sculpture which is not contained in the stone. The stone has no sculpture, the stone is a stone. But the hand of the artist uses that stone to create this sculpture. So chemistry uses the components of matter to create new forms of matter. So I would say chemistry is the art of matter. Now, this possibility of creating an infinity of things has already been sensed quite astonishingly 500 years ago by Leonardo da Vinci. You know, all of you know the Gioconda, all of you know famous painting, which all the people in the world want to see. And he wrote, but that was fantastic painting, sure, but he wrote a sentence which for science and for us is, I would say, even more important. It's not as nice because the painting is fantastically nice, but the sentence is extremely powerful. So remember that Leonardo da Vinci was a painter, an artist, he was a scientist and an engineer. So this is a very strong combination. So what does he say? For those of you who read Italian, you can read the top. In English, where nature finishes to produce its own species. We are part of that, what natural things. Man begins using natural things. What is it? We now know these are the elements. These are the periodic table of the elements, which are our bricks, the bricks of matter, in harmony with this very nature. These are the laws of physics. You cannot do things against the way things can combine. But then the ending is fantastically strong for an artist transferring this thinking to science, to create an infinity of species. This opens the doors to all the combinations, to everything, and to an infinity of entities which don't exist yet. There is only one which has been produced. Okay, on other planets, which have living organisms, there may be other combinations, but it is possible to create all these which don't exist yet. Now, just a little bit of history. This fellow on the left-hand side is Prometheus. He's one of the gods of the Greek mythology. And Prometheus stole the fire of knowledge from the gods to give it to mankind. Here you see him running away, looking over his shoulder and having the fire of knowledge here. And he was sort of thinking, hey, the other guys are going to run after him to catch him, but they didn't catch him. He gave it to mankind. And here is Prometheus handing over the fire to mankind. But you have also to realize we cannot give it back. What we know, we know. We cannot erase it. We cannot go back. We know. This cannot be given back. People who say we will go back, we stop, it's not possible. What we know, we know. And of course, it will continue. So our path as living organisms on this planet, like human beings, leads us from the quest of knowledge to the control of our own destiny. We now can take into our own hands what evolution, spontaneous evolution, did by itself. we have now the power to do it. We have to be extremely careful, but I'm convinced that we will do it. In 1,000 years, I'm convinced we will not be the same. This can't be discussed. Usually that leads to a lot of discussions. So this is somebody you must know. Han Yu, 8th century. And this is for innovation. The title of your meeting is also innovation. And he says something very important for research and innovation. When you sit at the bottom of a well to look at the sky, you will find it very small. So stick out your head and look around. And science helps you to stick out your head and to act. In the past, we transferred science, research, knowledge by the ape to mankind. Then in present times, we do that by education, by transferring our knowledge from professors to students, then also to the general public and all this. So what happens in the future? Maybe this. This doesn't mean, I don't want to say by that we're only shaking hands with robots. I just want to say that we will transform ourselves, and at some stage, we will shake hands with another hand, which is not a natural hand. In fact, we already do it, because some people may have lost their hand, they have a prosthesis. We can still shake hands, but it's not a natural hand. Some of us have implanted teeth. Some have new lenses in the eyes. Others have some piece of titanium in their hip. This is transformation. We are already transformed. And let's push it to the end. If one of us gets a heart transplant, that person cannot say anymore to his spouse, I give you my heart. It is not their heart. But the important thing, as you realize, is the brain. The rest is pumps and structures. And that's what makes us different. So let me finish with brain, with mathematics. Pure brain, if you want. David Hilbert is a very famous mathematician who is buried in a famous university town in Germany, in Göttingen. And here is his tombstone. On this tombstone, I've written 2 sentences which he pronounced in one of his lectures. They're on the bottom here, the 2 sentences. What do they tell us? Wir müssen wissen. That's the one on top. We must know. That is what drives us. We must know. Okay, we're doing research. We must know. We want to know, and so on. What is the second one? Wir werden wissen. We will know. That is the strong conviction which makes us go forward. We are convinced that eventually we will know. So science is here directing the game. We will know thanks to science. And all of you, but of course, the present-day audience, it doesn't need to be convinced, but I'm quite sure that all of you agree that science shapes the future of humanity. And we all have to participate. Most of you, all of you do it already. So these are you also when I talk to general public, but that's what I would like to transfer as a message. And furthermore, another characteristic of science, which I find is in terms of human relationships as important as the rest, is the fact that science goes around the world unhindered, no borders, no political systems, no religions, nothing like that. Science has without borders. And as scientists, we are fantastically privileged. If I come to one university in China, I can talk to my colleagues there, no borders. If you go to New Zealand, you talk to scientists there, no borders. So this is certainly one of the major characteristics of science. All right, so I have presented you just a story. It's just a story. But I hope there are some important lessons one can take out of it, which are obvious in some ways, but it's always good to repeat them because too often people forget these basic lessons. Thank you very much and good luck to all.
Thank you so much, Professor Lin. This is wonderful. I personally enjoy your speech a lot, and so are our audiences. So we have a lot of questions in the Q&A box, but because of time, I can only ask one question on behalf of our audience. So the question is, what is the current state of supramolecular application in drug discovery, in commercialized drug discovery? And where do you think the innovations can be made in the upcoming years?
You mean in drug discovery? Okay, no, the people who discover drugs have not waited for the definition of supramolecular chemistry. Drug discovery exists a long time, but it was done, let's say, sort of empirically. It is said that in the Middle Ages already, when people had a headache, they were taking leaves from a tree, salix, and out of that they made a tea, and that tea had what is it? Salicylic acid, which is aspirin. So this kind of thing is really something which already existed. But of course, the knowledge and also the computations which have been done, very involved computations to calculate the shapes, the interactions, they lead to much better understanding of drug discovery. And all the companies do it. The big companies now have a huge set up, usually division, where they try to analyze the drugs to see what key you have to build for a given lock. But of course, you have to know the lock and the key. You cannot build the key if you don't know the lock.
Yeah,
but
absolutely.
I could elaborate on that later, but we don't have time now, because somebody else is coming up.
Thank you so much, Professor Lin. This is wonderful. Thank you so much for joining us today. And it's really, really nice to meet you today.
It's my pleasure. I would like to say just it's a very good thing that you have these 8 universities getting together, but there are other universities around the world. And I think you have to look into other regions too. All right, good. Bye-bye. And good luck to all of you. Okay.
Bye. Thank you so much. Thanks. So thanks for all of our audiences for joining us today at our conference. Our next session is starting in a few minutes. Please stay tuned and hope to see you again soon in the future. Thanks a lot.